Today I'm going to be talking about X inactivation, X-linked inheritance, and bar bodies. I will also be discussing how these factors play into particular phenotype in cats. To begin with, we have the Lyon hypothesis. This hypothesis was proposed by Mary Lyon in 1961. The hypothesis states that one X chromosome in each female cell becomes inactivated. The X that becomes inactivated is random and varies from cell to cell within the female's body. Branching off from the Lyon hypothesis, we have bar bodies. Bar bodies are simply the inactivated X in the cell. Bar bodies can also regulate the amount of an X-linked product that is being transcribed. Our last definition that we need to talk about is X-linked inheritance. The simple definition is that the gene causing the trait or the disorder is located on the X chromosome. So you may be wondering how X inactivation and X-linked inheritance are involved with each other. The answer is simple. X inactivation is a product of X-linked inheritance. The tortoiseshell phenotype in female cats is an X-linked trait, and X inactivation is present in all females. The only way to achieve a tortoiseshell cat is to have heterozygous alleles. It's also important to note that since there is an X inactivation, the cat will show both black and yellowish-orange pigment. So here we will be crossing a tortoiseshell female and a yellow male, and we see that the big B is a black pigment and the little b is a yellow-orange pigment. The female has both because she's tortoiseshell. So then we come down to the cross and we see that our progeny consists of a fourth tortoiseshell female, a fourth yellow female, a fourth black male, and a fourth yellow male. After seeing the previous example, you might be asking yourself, how does X inactivation cause a tortoiseshell cat? Well, the answer is quite simple. Big B is inactive in some cells, meaning the little b is active, while in the same case, the little b is inactive in some cells, meaning the big B is active. In the long run, the clusters of cells with the large B active and the little b active will combine to give the phenotype. For our second example, we will be considering a second trait, hair length. For this example, we took a yellow short-haired female and crossed her with a black, long-haired heterozygous male. To get our results, we made two Punnett squares, one based on color and one based on hair. After getting our results, we can now make a branch diagram. The branch diagram can be divided into three traits: gender, color, and hair. Multiply the traits by each other to understand the probability of receiving that type of cat. Lastly, I want to talk about why cloning a tortoiseshell cat would not result into another tortoiseshell cat. To clone a tortoiseshell female, a DNA cell would be taken out of the cat and used for the cloning. If you remember that X inactivation is present in all females, only one gene will be affecting the cell. With the clone cat only having one gene, it will not look like its mother. It will be black or yellow.